Hi, everyone. Kit knows that it triggers me to get started late. Drives me nuts. But I totally get it. Computers crash. So, Mogli, I have presenting rights, correct? Okay, I will present. Maybe. Leah, give me a thumbs up when you can see that. Excellent. All right, everybody. So I'm going to be quick tonight and also want to acknowledge that there's a bunch of people in the room that are brand new beginners and a bunch of people in the virtual room who are intermediates. So I have to kind of span the gap there, but we are recording these meetings so that people can go back and watch the presentation over and over if you'd like. And I'll give you a preview today on the lesson is you're going to want to do a screenshot on a couple of these slides for reasons that will become obvious later. So tonight we're going to do a quick space management review and then talk about summer Varroa monitoring. So as a reminder from last week and last month and as review, in order to keep your bees from swarming, you really must stay ahead of both the nectar storage space and the brood space to prevent that swarming. So as you can see from these 4 colonies, it really is a tale of 4 cities. And it's a couple of years ago, me managing the space that each of those colonies needed. Some of them only needed 2 deeps. One of them didn't even have a honey super on. Rather, they had a feeder. And the big one obviously has, let's see, 1, 2, 3, 4 supers on the top, 1 medium on the bottom that's just empty space that I didn't have any slatted racks and they were bearding pretty badly. So I just put a medium on the bottom with undrawn comb just for the extra bees to hang out when it was hot. It was hot that summer like it is right now. And as you will also notice, I have upper entrances so that the bees can have easy access to the nectar storage area. If they have easy access, then they can make more trips in a day. More trips in a day means more honey. So in order to keep the brood area not congested, you need to continually create space for the queen to lay more eggs. So that if you're using mediums, that's really easy. You can just add an extra box with drawn comb under the honey super, but above the brood. If you have deeps, it's a little more complicated, unless you wanted to add a 3rd deep, and most people don't. So you need to either remove frames of capped honey and give them an empty frame to draw wax, or if you have drawn comb, add that. Or you can give frames of capped brood from your congested colony to a smaller colony and take the drawn wax that's empty from those smaller colonies and put them in your bigger colonies. So if you think about it, a queen can lay about 1 frame of eggs in 2 days. So you'll need a couple of empty frames for her to lay on. And remember that some of the bees are emerging too, so they're continually freeing up space. If you don't leave enough space for the brood and for honey storage, what they'll do if the honey storage area gets congested is they'll start filling the brood space that you just created for the queen to lay in. They'll fill that up with nectar instead, and then you get a congested brood area again. So I check my colonies once a week. When it's hot like this, it kind of sucks in the afternoon to do that, but it's really important to stay ahead of that swarm urge by managing that space. So as far as the honey storage, this is a review as well. You can go ahead and checkerboard the honey storage area, and that's literally just like a checkerboard. You see I've got 5 frames in there that are already drawn comb that have honey on them, and then I have 4 frames, and then there's a 5th one that's out of the camera view that you can just barely see the tab on. So I've got 5 and 5. And then if you've got another box, you can do the opposite. So you have a drawn comb over the top of an empty frame. And they really hate that because they're a little OCD about their space. So that'll help slow down the swarming urge too. Just a quick reminder, don't do that in your brood box. It's okay to put, 1 frame in between the brood on either side, but don't checkerboard it all the way across. The bees really hate that, and it tends to make them more easily swarm than otherwise. Okay, so this is the slide that you're going to want to take a screenshot on because I'm not going to go through these methods. But so starting now, you should be monitoring for Varroa monthly. I get questions, and I've seen it on the blogs a lot. Hey, what's everybody using to treat? And people text me and ask, I need to treat my bees. What should I use? My first question is always, what's the mite count? And so I don't treat any of my colonies, and most experienced beekeepers don't just automatically treat for Varroa. Rather, we do either an alcohol wash or a sugar shake to first determine, do they even need treating? Because there's no need to treat if it's below 2% mite. And don't think that you can look in your colony and see the mites, because most of the time, unlike this poor unfortunate bee with deformed wing virus and the mite on the top, 90% of the time the mite is on the underside of the bee, so you don't see them. So I really believe that an alcohol wash is the most accurate and effective way to measure for Varroa. Some people don't like to do that because you do sacrifice the 300 bees, but I figure that if you sacrifice 300 bees to save 50,000, then the bees would happily invest that as well. So the links for how to do an alcohol wash and how to do a sugar shake method are both available here. The other place that has a wealth of information, if you want to nerd out and read an endless supply of information about mite monitoring and how to do it and which is more effective than the other, take a look at Randy Oliver's scientific beekeeping website. He has nearly an endless supply of information that is scientifically based and accurate that you can spend hours and hours reading. Now, if you don't want to do an alcohol wash or a sugar shake, doing a sticky board 24-hour count is the next best thing. And a general rule of speaking there is if you have more than 8 frames of bees, that you should probably treat if there's 40 or more mites on that sticky board. And then if you have a small colony of 7 or less frames of bees, then treat if there's more than 20 mites. And again, that's a 24-hour count. And don't forget to make your sticky board sticky by spraying some PAM cooking sprayer, an equivalent on the board so the mites don't crawl off. One other thing about mites that I don't have on here, but it's important to sample from the right place in the colony. And so the place that you're going to find the highest concentrations of mites are on those cells that the bees are just getting ready to cap. So it's big larvae, and some of them are already capped. And the reason that is because the mites are getting ready to jump into those cells to start reproducing. So they'll be concentrated on that particular cell. And the best way to do that is to take that frame out and have a half cup measuring cup with you, and either your sugar shake bottle or your alcohol wash bottle with you. You take the frame of bees out, check it for the queen, because you don't want the queen on there, especially if you're doing an alcohol wash. And then what you do is you take that frame and you hold it over a bucket, and you just smack the frame down on the bucket. Once the bees are in there, a whole bunch of them will fly up, and that's okay because those are the foragers anyway. It's the nurse bees that are crawling on the bottom that you really want. So just give your bucket a shake, take your half cup scoop out, and that's roughly 300 bees. And you dump that in either the sugar shake bottle or your alcohol wash bottle. And then you shake, 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 shake. If you're doing the sugar shake method, you have to shake it for a good long time. The alcohol wash, you can shake it for 15 seconds or so, and that's plenty. And then what you do is count the mites. And if you're super scientific about it, you can count the number of bees that you have. If you've done an alcohol wash and figure out, oh, I had 310 bees or whatever, and then divide by the number of bees by the number of mites you have, and that gives you your percentage. Or the sugar shake, you have to shake it. I think it's at least 30 seconds for each one. And then you count the mites that come out. So there's your two methods. And I think I have a homework assignment for everybody. Since we're not presenting in July, our next meeting isn't till the end of August. And August is typically a dearth. And I suspect that July this month with this hot may be a dearth as well, as soon as the blackberries dry up and or stop blooming. So Randy Oliver has a great presentation called Reading the Combs. And so your homework assignment for July is to watch his presentation. All you have to do is Google it, Randy Oliver Reading the Combs, comes right up. And then you'll want to pay attention to that, because then during August especially, you'll want to really watch your combs and watch for pollen, because oftentimes the queens will stop laying in August, and August is a key time that they actually start raising winter bees. So you want to make sure that she doesn't stop laying eggs. And so oftentimes during August, we need to supplement their food with pollen patties. And so I'll leave it at that and see if there are any questions.
We have had a couple of questions in the chat. Do you want me to read them to you, Tracy?
Yeah, do you mind? I've got my iPad.
Let's see. So one question was not exactly related to this, but it was a question about whether you needed to brush off the bees when you take a frame from one hive and you would want to put it in the other hive.
Yeah, great question. So just make sure the queen's not on there. And I usually take capped brood. So capped brood almost always just has nurse bees on it. So if you take that frame out and you just give it a light shake over the top of your colony, the nurse bees hold on for dear life. And so just give it a little shake, and the foragers jump off or some fall in, and then you've got fewer bees to look at, and you look on the frame to make sure there's no queen. She's usually not on that frame anyway, because there's nothing for her to do there. So then you take that and you can put it right into the colony, into the smaller colony. The nurse bees will not fight. Now, if you've got the queen on there, a fight will ensue. So just make sure the queen's not on there.
And then we have a question about where to buy Varroa mite treatment. Any thoughts?
Where to buy Varroa mite treatments? Is that the question?
Yep.
So all the beekeeping supply places have it. Amazon's not my favorite place to buy beekeeping supplies or Varroa treatment, because the prices are a lot more on Amazon. I've done double checking, and I'm sure they have Prime delivery, but if you can wait for a couple days, sometimes it's so much more expensive on Prime. It's cheaper to pay the shipping. So I would definitely look at the beekeeping supply places. I like Man Lake. They deliver within a couple days from California, but there's lots of places you can get it. And to watch the temperatures. I know that's not part of the question, but it's so warm right now. Each of the treatments have a temperature range that you're supposed to use them in, and not supposed to use them outside of those. So especially Formic Pro, if it's too hot, especially the first 3 days, you can kill a lot of bees and your queen. So just make sure that you watch the weather forecast and choose accordingly.
Hey, Tracy, I have a question. This is Jeff.
Go ahead, Jeff.
Okay, I actually have there are kind of a couple questions. They're all related to swarming. I'm in my 4th year now, and every year I've had a swarm so far. Um, last couple of years I've been able to catch them. This year, so one hive overwintered, wasn't sure, so I bought 2 nukes. I started out with 3. I just split my last one into an 8th nuke. Now I have 8 hives in my backyard right now because I cannot figure out how to manage the frames. So so my questions, I've tried a bunch of different stuff now over the years. I haven't switched my deeps in the middle of the process. I've done it when in the beginning when they're too full. I put the empty one on top. On the one that I just found the beginnings of queen cells, they're not filled with anything yet, but there's about 5 or 6 um, cells that are facing down, but they're not the size of a queen yet. They're not huge, but I'm imagining that they're going to fill it and they're going to spread them down.
So is there anything in those, or are they just queen cups? Because once the queen lays an egg, then they become a queen cell technically. And if there's just a little cup there and there's no egg or larva, it's a queen cup.
They're just cups so far. Okay. So my main question on this one, and I noticed in my last notes that they're running out of space, but they've got 2 deeps. I pulled the feeder a little bit early because I left town. I put the honey super on top. So they haven't filled it because the nectar flow started later than I thought. But they haven't really adapted to the fact there's a super up there and they've run out of room. And this is the first inspection a week later that I saw the queen cups. So I'm wondering, is there any way I can prevent a swarm on that hive?
Sure. So there are no absolutes in beekeeping.
Yeah, right.
So let me ask you, are there any drawn combs in that super?
You know, I had a note that said that they had, but when I flipped through it tonight, like the records, I didn't actually see any. But I think that they had started drawing comb earlier, but they didn't do much with it since.
And do you have a queen excluder on?
Yes.
So the first thing I would do is I would take the queen excluder off because they don't recognize foundation with undrawn comb as space. That's where they're filling your brood boxes with honey. Okay. And they won't go through a queen excluder. Well, they usually won't go through a queen excluder to draw comb on an empty box of foundation.
Oh, wow. Okay, very cool to know.
Yep.
Okay, great. Is that it?
That will help. If you have any, do you have any supers with comb, any frames of comb that are drawn anywhere?
No, because I've been so swarmy over my thing. I've never really had a good honey flow. Okay. So I've never gotten a lot of honey super full of honey.
Okay, so if you have any frames of drawn comb or half drawn comb, you can put 1 or 2 in there and that will help draw them up in there. But take the super off for now. And then once they draw it out, the queen might start laying eggs in there, but you can always put the excluder on later. And then just those few will hatch and they'll backfill all that with honey.
Okay. Thank you. As all this was happening, I was watching the frames fill. I noticed, okay, there's some frames that have the honey in the top, then they get the pollen next, and then they get the brood. So, and I know, okay, you can kind of time it out when that brood's going to hatch. They're going to be available again for either honey or more brood. So right. What I've noticed in several of my double boxes is that the queen has filled every cell, and then the honey's coming down, and the brood is kind of switching. And I'm not very good at eyeballing where she's going with it and what her intentions are and how to keep her space available. Because again, I got a double box that's filled 3 times now in 3 different hives.
Well, you can take those outside frames that are all honey and you can extract those if you haven't used mite treatments on that comb.
Okay, leading into my next question. If I have fed them heavily sugar water, is a lot of that drawn honey actually going to be my own sugar mixed with pollen?
Yep. But it's edible. Just don't sell it.
Got it. Okay. And it's better than them swarming.
Yep.
Okay. Right. Oh, okay. Thank you.
You bet. Erica, I see your hands up.
Oh, hi. Can you hear me?
Yes.
Okay, great. Yes, I just have a related question, and sorry, I missed the presentation, so I completely apologize if I'm asking something that's already been answered. But I have a similar situation where a few weeks ago we found queen cells with eggs in them and starting to be filled with royal jelly. We decided to cut them all out, or I asked a beekeeper for some advice, and he recommended cutting out all of those queen cells and then adding a super on top, which I did. I actually had a super with empty but drawn comb. So I did that, and then we just did an inspection today, and there's already lots more queen cells that look like some of them are already fully capped as far as I can tell. And it looks like I maybe have some supersedure cells. And I only have this one hive, which started as a brand new nuke back in like end of April.
So the best thing for you to do right now is to go out there and split the queen out. So you can find the queen, get her out of there.
Yeah.
And there's an infinite number of ways you can do that split, but you want to get the old queen out of there, and if you have to, put them in the nuke box that you have, or start a second colony. It's always good to have 2 colonies going into winter, so you could split some of them out, start a new colony, and then what you want to do is you want to smash all but 2 queen cells. Leave 2. Yeah, if you leave a bunch, then they'll swarm and they'll swarm and they'll swarm and swarm again.
All but 2 queen cells and supersedure cells, or just
all of them? You only leave them 2 options. That way they have 1 queen and a backup.
Okie doke. And so for this current colony, it's 2 deeps. Should I just do like 1 and 1? Should I just like take 1 deep and move it over and then put
if your goal is to get some honey, then I would leave as many bees as possible and just take a frame with honey and pollen, and whichever frame the queen's on, that's easiest. Take those 2 frames, and then also a frame of capped brood.
Yeah.
And put those 3 in the middle of a deep box, and then put whatever other comb and undrawn frames you have in there and start a new nuke.
Okay. And then should I combine those hives like before the next nectar flow, whatever that is? Or
well, blackberry's it. So so the best thing to do if you want honey is to leave as many as you can in that big colony, and then after the honey flows over, then you can decide, do you want to go into winter with 2 colonies or 1? You can always recombine them later.
Okay. All right. Perfect. Thank you.
Tracy, if I can jump in with a question I was asked a while ago from Nick, is how long would you recommend waiting on new nuke or splits to check for mites? Can you check right away, or do you have to wait after you split them?
Oh, it depends on how big the split is, right? If you have a little tiny nuke like I just proposed Erica make, then I would wait, at least a month for them to get, a bigger colony. And if you do a sugar shake, you can do it right away because most of those bees live. It's just not as accurate. Kit, I see your hand's been up for a while. All right, we're gonna go with.
I couldn't find my mouse. Anyway, what is the story? Why won't they draw brood comb above the excluder? Honeycomb. If they need honeycomb, I understand why they wouldn't build brood, but
they don't recognize that as space, and they're not going to go up there to squeeze through an excluder to do it.
What if it's among drawn frames?
Drawn frames, they'll do it, but not.
No. Will they draw it? Will they draw a plastic frame between two above the excluder? No.
They hate it.
Fascinating.
I mean, if you have a frame with honey on it already, even if it's only a little bit and some drawn comb, they'll go up there and do that. But it's a lot easier to get them to draw comb, especially if you're not feeding them. If you take the excluder off and then put it back on after they get it drawn.
Yeah. and the queen meets them right in this moment and lays up the whole gorgeous thing. And no, I get it. Okay, thank you.
Yeah. Okay. I don't know your first name. S. Johansson?
Hi, thank you. I was messaging you earlier, Tracy. It's Michelle. I'm on my
Oh.
daughter's Teams app for her school. So I am a first-year beekeeper and totally got behind in my nectar space. And so I've got a queenless hive and every frame, except for the ones on the very outer edges, is either honey or nectar. And I am leaving town on Saturday and I'm gonna be gone for 10 days and I don't know what to do at this point.
Yeah.
I have a honey super on there that they're just not interested. They're not going up there. No excluder. They're just staying in the 2 deeps. What do I do?
So we can talk more tomorrow when you pick your queen up, but
Okay.
we can all, I mean, I would move the frames you have that have open space down to the deep, and then put some, if you have extra foundation, put those down there too, and that way she'll have something to lay on, and you know, a place to put the queen in the cage on that drawn comb, and then there can be undrawn comb on the other side, but I'll help you with that tomorrow.
Okay, thank you.
Yep.
We have a question from Raina if you want to take it.
Sure.
They came in. Raina, do you want to unmute?
I was wondering what was the link for telling whether or not your bees are hungry?
Oh, Google Randy Oliver reading the combs.
Okay, thanks.
Yep.
Perfect. Next we have Lisa. Lisa, do you want to unmute yourself?
Yeah, hi there. I have a question, actually 2 questions now. So today I did my hive inspection. In anticipation of the hot weather we're having, I created a small crack between the 2 deeps for ventilation, because I thought that's what I've done for years. And then I started, I better check everything I've been doing for years, right? Because you think you know everything, and then you find out I've been doing it wrong the whole time.
Hey, I still do that. I'm like, what? Oh.
So I've been doing really thorough hive inspections. I got a checklist. I'm doing it every week. It's actually quite fascinating. I did see some bees hatching because I've actually spent the time looking through them, and
That's really fun.
That was really cool. So, but the question is, what about ventilation with the hot weather coming?
Did you see my picture before with the upper entrances?
Yeah, I did.
I'll present and show. I'm using my mouse with trying to get my computer to do my laptop. Oh, here we go. Loading. Easier with a picture. So notice where all the bees are in the middle. I use queen excluders. Some people love them, some people hate them, but I use them. And you see where all those bees are. I have shims called emery shims around right above my excluder, and then every 2 honey supers. Plus, I have notched inner covers that I use all year long.
Oh, wow.
And I use screened bottom boards, so that offers plenty of ventilation. And the other cool thing about having all those upper entrances is it helps create a chimney effect, so that warm or all that hot air blows up just like out of a chimney, rises through there, and it helps dry out the honey.
Oh, okay. So
I don't recommend using a full stick to prop the super up, because then that leaves a lot of space that robbing bees and later this summer that yellow jackets can get into. So that's why I only use these shims, and ever since I've started using them, it really helps increase the honey production, and it helps dry it out faster. Plus, when the yellow jackets get nasty, they can they can defend those openings.
So are the shims all the way on all four sides?
Yeah, all the way around. It's just like it just is. I just use scrap wood, but you can actually buy emery shims. It's IMIRIE.
Okay.
It's just the width of a box.
Okay.
You know, the same width as the lumber. Some of mine are thicker because I use scrap, but anyway, and then you just have a couple inch notched opening there.
Got it. Okay. And when you drill a hole, you said where the honey is, how big of a hole would that be?
I do a 3/8 inch deep hole, and then I do it a couple inches wide.
Okay.
I usually just take my skill saw and start zipping it, set it to 3/8 of an inch.
Okay, I can do that. The other question came up. You just mentioned that blackberry is our last nectar flow, and I was knotweed, maple, blackberry, and knotweed.
Well, so knotweed, depending on whether you have it in your area, but blackberry is the one major flow that we have. So the knotweed will come on if you're near the mountains where there's fireweed, or along the railroad tracks too. In a lot of places, there's lots of fireweed. So knotweed and fireweed are both fall. And then ivy too, but that's really late and that's not really usable honey. So, but blackberry, if you want to count on getting honey for you, the blackberries is what it's all about.
Got it. Okay. Thank you so much.
Kit, how are we doing on time? Do we have time for one last question?
Yeah, we do. One more, and then I'll take over. Thank you for asking.
Okay.
I think, Kit, from what I can tell, you were the next question.
Oh, good. Okay. Well, that takes care of me. I have another question though, Tracy. My experience with emery shims is that they go crazy drawing comb in it. It wasn't my favorite experience.
They can draw comb in those. What I did an experiment and I looked to see if I do the notch facing down versus the notch facing up, how much comb they draw in the middle. And I don't know the answer why. Don't ask me why, because it makes no sense whatsoever to me, lowly human. But when I put the emery shim openings up like a U, they don't draw as much comb in it.
I'm sure I put it down. So, okay, I've got to give that a try.
I don't know why. But you know, if they draw comb and fill it with honey, I just scrape it out and stick it in a Tupperware bowl, and my daughter eats it like candy. So,
yeah, my solution was to cut the holes in the bottom of every other medium instead of using the emery shims. So I cut a 3/4 inch by 3/8 hole at the bottom of each using a jigsaw.
Yeah, you can do that. I don't like to have to plug it later, so.
Yeah, they'd probably plug it with propolis for the winter time. That's not a bad thought. Or a circle hole in it that you can plug with a cork.
Yeah,
I've seen that done.
Yeah, if you do that, just make sure you don't put it anywhere near your handles for obvious reasons.
Okay. Thank you. Kelly, are you with us right now? Kelly Kulhanek?
Yes, I'm here.
Oh, super. Great. Welcome. Okay, I'll introduce you in a minute after a few little comments. So I am president of Puget Sound Beekeepers Association. I'm Kit Hyatt, and just a word in case you haven't heard or been to a meeting before. We are a 501c3 or a charitable organization, and essentially what that means is that we are all volunteers, no one is paid, and everything that you see happens because people are, enjoying doing it. And so if there's something that you feel like you might want to be contributing, please reach out and get involved. There's odds and ends things as we come up out of remote meetings. There will be some more activity for those who who are volunteering. It's been a little challenging to keep people involved without people to be connecting to, you know, events and so forth. We do have need, as you may have seen in the newsletter that I sent out yesterday. Our volunteer who's managed the extractor rental for the last couple of years, Margaret Stein, has resigned. And while she's open to kind of transitioning over, we do need to find someone else to fill her shoes. And that means that someone would meet at the apiary there at the Arboretum and greet people, you know, unlock the facility to get the extractor out and give it to the person who has signed up and paid for it. So then they need to meet them a week later and take it back and maybe meet another person who's renting for the following week. It happens in clusters. It isn't super busy with that extractor rental even before COVID. So anyway, if there's anyone that lives in the area of Capitol Hill or the U District, it would sure be awesome to hear from you so that we could, you know, talk further about it. And thanks for that thought. Next month, as Magali said, Tracy said there's no meeting. And so we're gonna watch Randy Oliver's YouTube. I love that idea. We are, however, having our annual picnic at the apiary, which is behind the main building at the Arboretum, if there are people who haven't seen our apiary yet. And there's a covered area with barbecues, and everyone brings wonderful food, and it's always been a really special gathering. So if people would like to be able to join us, it'll be on a Saturday afternoon. I think the fourth Saturday of July. So look for information about that to remind everyone and consider joining us. And we are working to be able to present the meeting as a recording in addition to the live when we go live, so that people who expressed needs of staying home with kids and not coming way up from the South End. And I mean, these meetings kind of spoil us in a way, although I just can't wait to see people. So just know that we'll have it be dual. It'll be on a two-way deal. And I know that a lot of people watch the YouTube issue of each meeting, and I think when Tracy lobs off so much information that I can't quite catch it all, or I just want to learn it twice. You know, it's really nice to be able to go back and hear. What else do I need to share? The link that we have for the meeting used to be unique each month. And now that Magali is mastering the deal, it's the same link. So if that reassures anyone to know that they've got the link for the meeting, it's easy. So the apiary, I can mention that we're working, the hang up with the apiary is that everyone needs to have a bee suit, and we provide bee suits. There are other clubs in Western Washington that are meeting, but people are bringing their own equipment. And a lot of people who come to work in the apiary and have that hands-on experience before they get bees is often the situation, and they don't have the equipment that they need. And they would need a bee suit, and it would be, so we're working on figuring out how to spray them with something, or what the requirements are to meet cleanliness, to have those suits as a backup to those who don't bring suits. So I do know, you know, we do want to open the apiary up because it's such a great way for people to learn. And that's really its purpose, right? That's why the bees are there in the first place. So we'll get that happening. And everyone knows about the swarm list. It has been really busy. There are a lot of swarms happening. It's kind of amazing, actually. And so I'll do our parts to keep our bees in our boxes is, of course, the best plan by checking for swarm cells on a weekly basis. Queen cups are just a signal, and you can scrape them off, and you can scrape it all off until it's capped, and then you're in deep trouble. So that takes 8 days for that to occur if you missed the queen cup. So a weekly visit is awesome this time of year. And extra equipment, which is not something that's really always on hand after you've already caught 2 swarms. So anyway, okay, so Kelly Kulhanek is next up, and she is with us from Washington State University. And I'm excited to learn about her research about how mites travel between hives, because it seems to me that mite populations vary a lot in hives that are right next to each other, but it may be my imagination. And so it will really be wonderful to learn what she studied, what she came up with for her PhD dissertation. And she's got other things up her sleeve too. But anyway, Kelly has just moved to Washington State from the East Coast. And she was going to come to Seattle and present with us in person. And I know she was excited to be able to come to visit Seattle. So maybe we'll have to do that another time. Anyway, Kelly, you ready? You can take over from here. Thank you for being here.
Thank you. I'm trying to share my screen and it's just giving me like a loading circle, but hopefully it'll work. It's just taking a minute. I have a little more experience with Zoom than I do with Teams, so I'm trying. But yeah, I guess I'll just start talking about myself while I wait for my slides to show up. So yeah, I did just move here from Maryland. I did my PhD at University of Maryland. I started there in 2016, and I finished my PhD in the spring of last year in April, like right at the beginning of the pandemic. So it was a weird time to sort of finish something monumental and move cross country and start from scratch, all in a pandemic. But we're happy to be here. I'm actually originally from San Diego, so I'm happy to be back on the West Coast and a little bit closer to my family. And this screen share is really struggling, it seems. Let me try it down here.
If it can help at all, Kelly, you can feel free to also send me your slides and I can share for you.
Sure, that might be easier.
Let me post my email in the chat.
Great, thank you. I might not have like the latest update of Teams or something, and sometimes that.
I practice before meetings to get used to it, and it is a little bit more of a hiccup. I'm sorry.
Oh, it's okay, no problem.
What I meant is even practicing doesn't always help.
Yeah, I'll just send that right over.
Tonight, where it was giving me the same thing, actually, Kelly. I was trying to get in and it was giving me I'm waiting.
Yeah.
Yeah, Teams does not fail gracefully. It just says it's just not doing anything.
All right, I just sent the email. Might take a minute. It's a large file, lots of photos.
I have it, so let me pull it up.
Great. But yeah, I've been here in Pullman since August, so a little bit less than a year. We just bought a house here, though. We bought this house. We closed with this house on Friday, so we'll be here for a little longer.
No, it's on mine that it's taking a little bit for downloading.
Okay.
Are you working at WSU?
Yes. So I'm a postdoctoral researcher, which is kind of like a residency after you get a medical doctorate degree. So you finish your PhD and then you kind of have these interim positions where you continue to do research and I'm still working under a PI. I work for Brandon Hopkins here. But you just kind of like build your CV while you look for a more permanent position, a professorship or a tenure track position. So yeah, I'm not sure how long we'll be here. My fiance, I'm pointing up because he's upstairs, is considering getting a master's here. So we think we'll be here for at least a couple of years. And I have been to Seattle a couple times before. It's absolutely gorgeous, especially this time of year. So hopefully I'll get to come out and speak with you all in person at some point. Okay, great. So I'll just, I guess, try to signal you for next slide. But yeah, here's my introduction slide. So I'm just going to give some updates on the indoor storage research that our lab is doing here at WSU. I work a lot here on commercial beekeeping management practices. It's a little bit more of a large scale operation here than we had in Maryland. In Maryland, I think the largest beekeeper in the state is like 200 or 300 hives. So it was definitely a little bit smaller scale situation out there and kind of leveled up to a more commercial situation out here. So working more on commercial management practices, including indoor storage, and I'll give an introduction as to what that is for newer beekeepers who aren't familiar. And then, as Kit mentioned, I'm also going to talk about landscape level Varroa spread. So this was one of the chapters of my dissertation that I did while I was in Maryland, kind of trying to understand how bees and mites are moving between colonies across the landscape, between apiaries, between your neighbors and your own hives, and things like that. So next slide, please. So I'm going to start with the indoor storage stuff. And I was told that there's a bit of a mix of experience in the group. So I'm just going to give a really basic introduction as to what this is. A lot of people on the East Coast have never really heard of it because nobody does it there. But the basic idea is that these commercial beekeepers move their entire or the vast majority of their operation into indoor storage rooms for the wintering period. And this is a picture of a facility just north of Othello, Washington, where a big commercial beekeeper there moves all of his hives into. And you can see it's just a dirt floor. It's actually like a potato shed. So some of these things are higher tech and lower tech, but as long as it maintains a cold temperature so the bees don't start flying while they're in there, then you're good to go. Next slide. I have a lot of animations, so a lot of the clicks are not actually going to change the slide, but it'll change the words. Sorry, go back. Yeah. So the idea here for the commercial beekeeper, the incentive for doing this is that they can reduce inputs into these colonies over the wintering period compared to outdoor wintering in warmer climates. So kind of the alternative for a typical commercial migratory beekeeper is that they might winter their colonies in Southern California or in the Southern US, where it's a little bit more of a temperate situation. It's not as cold, it's not as harsh. But in those warmer climates, the colonies can continue to produce brood, which means the mites can continue to reproduce. The bees will be out flying and trying to find things to forage on, but there's really not a lot blooming in those times of years usually. So the colonies require inputs, right? They require inspection and feeding and mite treatment sometimes. So the idea behind the indoor storage is that they can put their colonies into this somewhat temperature controlled situation where they kind of force this dormancy period to last however long they need it to reduce the inputs into the colonies. And next slide. And there are a lot of potential applications of indoor storage that commercial beekeepers are really interested in. So, you know, it's a pretty unique situation where all of their hives are in one central location and even inside one room. And it really presents an opportunity for a lot of other management practices and techniques that they could start to use. But this is sort of a more recent thing that's gaining popularity really just over the last couple of years. So as more and more beekeepers are starting to do this, they are getting more and more curious about different ways to maximize the potential of indoor storage. And a lot of the time those beekeepers call Brandon and they say, what do you think about this? You know, is this possible? Do you know anyone who does this? So in our lab, we try to test out a lot of these things before we recommend them to beekeepers, so we can make sure they work before beekeepers use these practices on, you know, their entire operation at once. Next slide. So a couple potential applications that we're working on that I'm just going to mention and not go into detail on are Varroa control via fumigation. So, you know, the beekeepers are super curious about, we have all our hives in one room. We have control over the atmosphere in those rooms. We control the airflow in and the airflow out. So a couple things our lab is looking into is trying to fumigate these hives for mites while they're in these storage facilities. There's a couple of ways we're thinking about doing that. One way is actually with carbon dioxide. So honeybees naturally produce quite a bit of carbon dioxide when they're clustered in the winter. The percentage of carbon dioxide can get up to 6%. And other studies have shown that mite mortality, Varroa mites, will die at around 2% carbon dioxide. So the idea here is that if we can get the CO2 high enough in these buildings to kill mites without harming the hives, then that might be a really good opportunity to control mites with less harsh chemicals and less input of actual pesticides into the colonies, and kind of give the beekeepers a bit of a jump start at the very beginning of the year. The other way to do this is with the more traditional fumigant that we usually think of, which is oxalic acid. You know, beekeepers think of the possibility of fumigating their entire operation with oxalic acid at the flip of a switch, instead of having an individual applicator go through and fumigate each hive individually. It would save them tons of time and labor and money. So that's another thing of interest. That obviously presents a lot of concerns safety-wise and environment-wise. So there's a lot of hurdles to jump for that one before we really get into doing that. But these are sort of preliminary things that are going on in the lab. You can click forward. Queen banking. So the idea here, and you may have dealt with this as a hobbyist beekeeper, is that the time of year where there is an abundance of queens available from commercial queen producers can be a little bit mismatched from when you need them as a beekeeper, right? Like, everybody wants queens as early as possible in the spring. As soon as it starts getting warm, people want those queens available, but that's the same time period where the queen producers are just starting to be able to produce queens and start things up because of weather restrictions. So the idea here is that indoor storage could be used to bank queens for longer periods of time. So bees or queens that were made in the fall could be stored indoors until the following spring, so that they're ready to go right when beekeepers start needing them a little bit earlier in the season, and kind of shifting the timing of when queens are available to better meet demand. So that's another thing we're working on, more preliminary data that I'm not going to show you today. But again, if I come back, hopefully we'll have a little bit more concrete results for that stuff. Next slide. All right, so the other idea that we have that we're working on is of the commercial scale brood break. So next slide. I'm going to go into more detail on this one. Click again. Thank you. So I'm just going to give a brief introduction as to what a brood break is and why they're useful, in case people aren't familiar. So basically the idea is that mites reproduce inside the capped brood in a colony. Go ahead and click. And the majority of mite population can be underneath the capped brood. You know, really, mites are spending a relatively short amount of time in that phoretic stage, and they kind of want to re-enter a brood cell as quickly as possible to start another reproductive cycle. So a huge amount of a colony's mites can be underneath the capped brood. Click again. And that capped brood protects mites from some chemical treatments. So some treatments can penetrate under that capped brood, but a lot of treatments that you apply to your hive don't penetrate under that capped brood, which is why you have to do it kind of 3 weeks in a row or leave it on for a total of 3 weeks, so that you clean out a whole brood cycle. And as the mites hatch out of the brood, then they're exposed to that chemical that you've applied. But that presence of capped brood at any given time really protects a lot of the mites from being affected by these treatments. And people use brood breaks to reduce the amount of capped brood that is present in the colony. So a brood break just refers to exactly what it sounds like, that beekeepers can sort of artificially manipulate the colony in some way to cause a break in brood production, force all of the mites in the hive into the phoretic stage, and then you can get away with a single application of a miticide treatment instead of having to do, multiple treatments in a row to clear out a whole brood cycle. So that is the idea here. Most of the ways we have to induce brood breaks are relatively labor intensive. So, they require either going into your hive and caging the queen so that she's confined to a single area and she can't lay. But that's pretty labor intensive to go through each individual hive and find the queen, cage her, and then have to come back again and release her a few weeks later. So commercial beekeepers have trouble implementing more labor intensive individual hive management strategies. So the idea here is to try to use indoor storage in the summer to help these beekeepers initiate a brood break with less labor overall. They have to move the colonies in and out, but they don't have to touch each individual colony in the process. Next slide. Yeah, so the idea here is that the colonies would actually go into these cold storage facilities in the middle of the summer, whenever is convenient in the beekeeper's schedule, usually probably right after honey production or right before honey production. Sometimes there's a little bit of gap in between pollination contracts. So the idea is that they would move their bees into cold storage for a period of 18 days to induce a brood break. And then they can get away with one Varroa treatment, or they get really good efficacy with a single Varroa treatment, so that the overall chemical inputs into the hive throughout the year can be reduced. Next click. So a master's student in the lab, and here's her little picture down here, Anna Webb, she just graduated this spring, worked really hard on this project, and she had set up this experiment where she had 2 treatment groups. So one treatment group is the indoor group, and there were 37 colonies in that group. And basically the indoor group was moved into cold storage, held at 4 degrees Celsius for a period of 18 days. And she had an outdoor control group that was at our new facility in Othello, Washington. And there were 20 colonies in that group. And basically what happened is she put the colonies in storage or left them outside for the period of 18 days. Then right after they came out of storage, or the corresponding outdoor colonies, they all received an oxalic acid dribble treatment. And then she measured the mite loads when that OA dribble was, 2 weeks after that OA dribble was applied. So the idea here is hopefully that the indoor storage would have greatly reduced the amount of capped brood present, and thus the oxalic acid would be a lot more effective in the colonies that had been indoors versus outdoors, because the outdoor colonies should have more mites still under their capped brood and not exposed to that single application of oxalic acid. Next slide. So the first result Anna found was really good. Here we're looking at the average frames of brood in each treatment group, indoor in the lighter color, outdoor in the darker color. And you can see before the storage period of 18 days and after the storage period of 18 days. And you can see both groups started with really similar frames of brood, right around 7 frames of brood per colony. And after storage, the indoor group had gone down to about 1 frame of brood per colony. So a really huge reduction in capped brood. The capped brood also went down a little bit in the outdoor group, and we think that's because there's really not much for them to eat in Othello, Washington. So we think, you know, they kind of came off of crop pollination where they had really abundant food sources, and then they got to Othello and were kind of shocked and kind of slowed down producing brood a little bit. This was in June and July. So the outdoor group lost a little bit of capped brood too, but the indoor group went almost down to no capped brood whatsoever, which is great. So that means that, you know, when we put colonies in indoor storage in the summer, it can help induce a brood break. Next slide. Okay, so then Anna put the oxalic acid dribble on, right? And so here we're just looking at the average mite load per 100 bees. When she put the oxalic acid on and 2 weeks later, and you can see that in both groups, indoor and outdoor, the mite load went down, but they went down at about the same rate, which is kind of surprising. And we have pretty similar ending mite loads here post-treatment. We would have expected the oxalic acid dribble to be a lot more effective in that indoor group because there was so much less capped brood present. And you can see that this red line here is the treatment threshold of 3 mites per 100 bees, which the relevancy of that kind of changes from season to season. But you can see that in either group, the mite load did not go down below that treatment threshold of 3 mites per 100 bees. So it seems like the oxalic acid just didn't really work that well in general. This was 2 weeks after application, and I'm kind of curious to see what it would have looked like 1 week after application, if maybe the mite loads would have been lower, closer to when the treatment had been applied. But kind of an interesting result. So more work to be done here for sure. Next slide, please. Thank you.
Hey,
So
Kelly, what concentration?
there is.
What
Sorry, go ahead.
concentration did they use? What concentration of oxalic acid did they use?
That's a great question, and I don't know the answer off the top of my head. I think it's whatever's typically recommended. I can look it up. I have it on my computer.
Okay, later. Thanks.
Yeah. Remind me later, please. So we still think that indoor storage has good potential for commercially applicable brood break options. The actual brood break portion did technically work. Just the efficacy of the oxalic acid didn't work as well. So next slide. Yeah, we applied for more funding to try this again and to try to figure out that issue with the miticide efficacy. We kind of want to scale this up to a larger scale with more hives. And see if we can figure out how to really fine tune this, because it does seem like a really good option to reduce the number of chemical miticides needed to kind of keep these mite loads low in these colonies. Next slide, please. And click one more time. So another big project our lab's working on right now regarding indoor storage is kind of addressing this issue that there aren't many large-scale long-term studies on the effect of indoor storage on colony health and survival. So beekeepers have been using indoor storage for a long time. It originated in Canada, like, 100 years ago, when beekeepers just needed to actually shelter their colonies from really harsh winters. But now that it's scaling up to such a massive level in the United States, and really being used by a lot of commercial beekeepers, we really need to make sure we understand if this is actually beneficial, if it's improving colony health, if it's improving colony survival, and if it's really economically viable for these beekeepers to be doing, because that is the bottom line. It has to work for their operation. So next slide, please. We were working on a big 2-year study in partnership with the Bee Informed Partnership, which was actually headquartered at my old lab in Maryland. So it was super fun for me to come over here and get to keep working with a lot of the same people on this collaboration. And we had 2 beekeepers participating in this study that started in August 2019 and went to March of this year, 2021. In each of those 2 beekeepers, we had 3 treatment groups. Go ahead and click. And 120 colonies total. And click. So in the first operation, which was located in Montana, kind of near Missoula, this beekeeper had 60 colonies participate, broken into those 3 treatment groups. So the first treatment group is the shed group, which is the indoor storage group. So these 20 colonies went into indoor storage for the winter. In purple, we have the outdoor north group. So those 20 colonies were overwintered outdoors in the north or in Montana or Idaho, where the beekeeper was based. And then the third treatment group, these 20 colonies were overwintered outdoors in Southern California, as is sort of the more typical option for commercial beekeepers. And then we repeated this again with a second operation in Idaho. Again, 3 more treatment groups with 60 more colonies, so 120 colonies total for 2 years. And go ahead and click. And these colonies were visited 4 times per year. And the actual timing and the months kind of depended on the beekeeper's schedule. But so I have sort of like their management timing and the month, just to kind of give you context. So the first visit was in August, and this is the pre-treatment visit, which was before the beekeepers had applied a fall Varroa treatment. The second visit took place in October, post-Varroa treatment application, and sort of right before, or as close as we could get without it being too cold, before the colonies went into indoor storage or were moved to California. Then we took a post-winter sample in January. So these are pretty much right after the hives have come out of storage and as soon as they arrive at almonds in California, very early January. And then a follow-up sample was taken after almond pollination, sort of at the end of February or the beginning of March. Go ahead and click. You can click 2 more times, I think. Perfect, thank you. So at each of these visits, we were inspecting the crap out of these bees and taking down sort of as many metrics as we could think of. So we were performing health inspections and recording queen status, brood pattern, frames of bees and frames of brood to really try to quantify, the colony size and the queen productivity and, colony size and brood area, especially going into almonds, is super critical for commercial beekeepers because that affects how much they can get paid for their colony rentals. We also took a lot of samples for Varroa, Nosema, and viruses. So all these different suite of viruses got sampled for as well. And then of course we recorded colony mortality if we found any also. Next slide. So I'm just gonna share a few results, and we are still waiting on a little bit more of this data. Some of the viruses, the viral samples are still being processed. But this is all like hot off the press preliminary data. So here we're looking at the Varroa loads in the first year, 2019 to 2020. And we have the pre-treatment sample all the way through the follow-up sample. And again, the sort of timing those correspond to is in the top right corner. And again, we're just looking at the average Varroa load per 100 bees. And you can see here sort of a weird thing happened at the pre-treatment sample where the purple group, the outdoor north group, started with a higher mite load than the other 2 groups. We try to control for that as much as possible, but when you're working with bees in the field and a commercial beekeeper's operation, sometimes you just can't help it and you don't know until after the samples have been processed. So that just happened to happen that first year. But then what you can see is at the post-treatment sample, the mite loads went down in both the outdoor north group and the outdoor California group. The outdoor California group actually has a significantly lower mite load at that post-treatment sample in October. And then the colonies go their separate ways. The shed group goes indoors, the California group goes to California. And then you can see that what happened at the post-winter sample, so this is from October to January here, the mite loads in the shed group and the northern group went down quite a bit between October and January, but they crept up slightly in the California group. And then the California group also crept up slightly again before that follow-up sample. So kind of interesting, that's kind of what I was touching on before, is that the concern with overwintering in outdoor California situations is that the bees continue to produce brood, which means the mites continue to reproduce, and those mite loads can kind of creep up slowly over this time period. It just so happened that that mite load was pretty low to begin with anyway, so probably not a problem for the beekeeper in this case, but still something to watch. And also you'll notice if you look at the y-axis that these beekeepers had super low mite loads anyway. The highest mite load here is 0.9 mites per 100 bees, so less than 1 mite per 100 bees, and that was even pre-treatment. So these beekeepers had really low mite pressure anyway. But yeah, next slide, next click, perfect. So the second year, 2020, 2021 was a little more interesting in my opinion. Again, you can see here we actually started all treatment groups the same. We did not get to the pre-treatment sample due to COVID and travel restrictions and things like that. So the first time we saw these colonies in the fall of 2020 was in October. But you can see that the outdoor California group, the mite load really increased quite a lot in the overwintering time period and was still pretty high at the follow-up as well, as opposed to these colonies that were indoors or in cold climates. So again, that kind of confirms our suspicion that these warmer climates kind of allow the mites to reproduce a little bit more and could be a little bit more of an issue. Next slide. So nosema, kind of a similar thing, and you can show the other one too for this. Perfect, thank you. You can see that the outdoor California group really starts to creep up in nosema loads post-winter and at the follow-up here in 2021. So a little bit more Varroa pressure, a little bit more nosema pressure. And another reason why we think that might be happening is that typically in these outdoor California wintering yards, there's a ton of colonies, right? A lot of the time this is like a holding yard situation with you know, triple digits of colonies kind of all packed into a small area, and they're flying and they're looking for things to eat and there's not a lot around. So there can be quite a bit of robbing in these instances, quite a bit of mixing of bees, drifting between colonies, which is a big opportunity for spreading mites and nosema, which I will talk about a little bit more in sort of the second half of the presentation. But that's another concern here, is that those bigger apiaries with lots of bees flying around could cause problems as well. Next slide. Click again. You can click through just to the rest of these bullet points. Perfect. Okay, so again, just to summarize, Varroa and nosema loads were increasing more in California post-winter. You know, we think this is because the bees are continuing to produce brood, but also that bees are flying more and mixing between other hives, to where these post-winter mite loads can definitely be a lot higher. Again, really low mite pressure overall, but we're also talking about January here where they should have pretty low mite loads. And if you kind of leave this unchecked, it can get worse and worse as the season progresses later on. So definitely something to think about. And we've actually applied for more funding to do this again. We want to keep looking into this because years 1 and 2 did look so different from each other. So yeah, we will keep sort of investigating the roles of this. But I do want to talk more about this idea of bees mixing between other hives. Go ahead and click. So I'm going to be talking about one of the chapters of my dissertation that I did while I was at Maryland, testing the role of horizontal transmission in rapid Varroa increases. So there's been a lot of work done in this area. This is kind of like a pretty hot topic, or it was when I was doing my PhD a couple of years ago. Maybe it's already old news. But there has been quite a bit of research on this idea of mites moving between hives within an apiary, but also between hives in separate apiaries. And we know that mites will move between colonies and apiaries. Click. And that crowding colonies and apiaries and in the landscape increases mite loads. So again, in like a holding yard situation where we have lots of colonies in a small area, there's typically more mite pressure in those colonies. Go ahead and click. It's also been shown that mites can immigrate into colonies up to 1.5 kilometers away, so quite a big distance. And go ahead and click. And there's a little bit of debate as to sort of the directionality of this event. And there's a little bit of question about like which way this is happening. So the first idea is that bees are dropping mites off at other hives. So you may have a high mite colony in the landscape that, you know, has a really high mite load for some reason. Maybe it hasn't been treated, or we all have that one hive sometimes where no matter what you do, it always has high mites for some reason. So, you know, one potential idea is that the bees from that colony are going out into the landscape, visiting nearby healthy or receiver colonies, as I'm calling it here, and dropping off mites in the process. And really kind of like going out and spreading their own mites to these other colonies in the process. And then the other idea is that bees are actually bringing mites home themselves. So say, you know, you have these really nice, beautiful, healthy colonies that have very low mite loads, but a dearth rolls around, you know, the honey flows over, they are going to start looking for resources wherever they can find it. A lot of the times that means they're visiting other colonies, participating in robbing, or just kind of snooping around looking for stuff. And in that process, it's pretty likely that the bees from that colony will find a high mite colony, enter that colony, you know, interact with the bees and mites in that colony, and they'll pick up mites in that process. And then when they return home, you know, they've picked up a phoretic mite, and then they bring it home themselves. And go ahead and click. So I kind of wanted to try to investigate if we could understand better how that's happening. And there has been work on this where people have looked into this. But usually it's on a relatively small number of colonies or a small number of apiaries, because usually this requires tracking individual bees, which is obviously very hard to do. But I wanted to try to do it at more of a realistic landscape scale over longer distances with more colonies and apiaries. So I set up this experiment, and this is a satellite image of the actual farm where I did this. It's a University of Maryland research farm in central Maryland. And basically I set up these 9 experimental apiaries, and we have one donor apiary at the center in yellow there. And then I set up 8 receiver apiaries around it. And you can see I tried to spread them out kind of in the different cardinal directions, north, south, east, and west. And I tried to have a little bit of a closer radius and a little bit of a further away radius to sort of get as much insight into this situation as we possibly could. Go ahead and click. So in the donor apiary, we placed 2 high mite and 2 low mite colonies. And each of these apiaries contains a total of 4 colonies. But in the donor apiary, we had 2 high mite, and those colonies had greater than 3 mites per 100 bees. I think one of them actually had like 10 mites per 100 bees. And then the low mite colonies had less than 1 mite per 100 bees. So a really low mite load. And the idea here is to try to track the frequency and the proportion of high and low mite bees ending up in all these receiver apiaries. So we can kind of get a better understanding of if these high mite colonies are the main driver of these, you know, really intense mite increases that we tend to see in the fall. We can hopefully kind of catch them in the act visiting these receiver apiaries and understand the effects of that. So in order to do that, go ahead and click. We had to paint all of the bees in the receiver apiary, and we didn't paint all of them. That's an exaggeration. I painted about 16,000 of them. So in each colony, we painted 2,000 bees, and I did it twice. So about a total of 16,000 bees. The red colony, or the high mite colonies, were painted red. So you can see this is a batch of bees from a high mite colony being painted red. And this is actually my fiance, who I dragged out to help me on a Saturday, painting bees with me. And we did this by anesthetizing them in batches with carbon dioxide. So we basically shook the whole hive into a plastic bin, and then took out batches of about 500 and knocked them out with CO2. And then we dump them out. This is just like an upturned sticky board here. And they're actually not very easy to paint while they're unconscious because they lay on their backs. But as they start to wake up, they'll flip over and they hold still long enough for you to be able to paint them. So we painted really kind of as many as we could. It was the majority of the adult population of these hives. They were relatively small hives, just 2 deeps or a deep and a medium. So again, the high mite colonies are painted red and the low mite colonies are painted blue. And go ahead and click. And then in the receiver apiaries, we had 4 colonies each. And, you know, the next step is to try to detect these painted bees in the receiver apiaries. And in the initial phase of this experiment, my advisor suggested that I just check each colony individually by hand, every single frame, every single day for these painted bees. And I tried that, and it turns out there are physically not enough hours in the day to make that happen. So I had to figure something else out. So what I ended up figuring out is that we could place these computer camera mechanisms on all the receiver colonies to actually detect the red or blue paint color. And whenever they detected that color, they automatically took a photo, saved it to the memory card, so I could just come by later, pick up the memory cards, and know if a red or blue bee had been there. And I just learned to do that on YouTube. I watched a lot of Python tutorials, so that was definitely a fun adventure and a good project. But I was really excited to have a solution where I didn't have to manually check every frame of every hive. So the cameras ended up being pretty cool. And you can go ahead and click. Another cool thing was that we were actually able to tell individual bees apart because we weren't that great at painting them. So this is an actual photo taken by one of those cameras at a receiver colony of the little blue bees arriving there and the camera detecting their paint color and their photo being taken. And you can see that each of these bees had a little bit of a different experience being painted. The one on the left is like a relatively nice circular mark. And you can see the one on the right, poor thing, got a little bit of paint on her head as well as her thorax. So because we were painting so many so fast and not taking as much care as you might in marking a queen, the sort of randomness of the paint marks allowed us to tell individual bees apart. And then go ahead and click. We also had red bees as well, of course, which is also easy to tell apart. And click. And then the last thing we added to receiver apiaries to kind of try to tease out how this was happening were robbing screens. Click one more time. So 2 of the 4 colonies in each of these apiaries were screened. And go ahead and click. And everybody is probably familiar with what robbing screens are, and you can click 2 more times. Perfect. But you know, I'll just briefly say the idea here is that the entrance to the colony is screened. The expected colony entrance, you know, that normal big opening at the bottom, still has all the smells and sounds of a normal colony entrance coming through because it's just a mesh screen. But the actual colony entrance is this very reduced entrance at the top of the screen. So the idea here is that non-natal bees who did not originate in this colony will go for that expected colony entrance, will be confused, and will not be able to gain access to this colony. And also that reduced entrance on top is harder to find and a lot easier for this colony to guard. So basically the idea here, by using the screens on half the receiver colonies, is that if these high mite donor bees were arriving in these receiver apiaries, the robbing screens should at least somewhat deter them from being able to enter these receiver colonies. And go ahead and click. So when all was said and done, this is what a typical receiver apiary would look like. You can see the little cameras mounted above the colony entrances in little Tupperware containers. It's just a Raspberry Pi motherboard, which is just like a really cheap, simple computer motherboard thing. They cost like $30. And you can use them to do all sorts of stuff. And then it has a simple camera attached, and you can see 2 of the colonies have robbing screens as well. So now we are ready to, set the donor bees loose, see where they go, in what frequencies, and see if these robbing screens do anything. And before I get into results, I just kind of want to walk you through what the expectations are of these 2 different ideas, these 2 different hypotheses we were kind of testing. Next slide. So this first idea is that the high mite colony bees are dropping mites off at other hives. So if that were true, next slide, we would expect to see a lot of high mite bees visiting these receiver colonies and not as many low mite bees. If, something about these high mite colonies is making these bees go out into the landscape and spread their mites around and be like this main driver of these rapid increases we see in the fall, then we should expect to see a majority of visits from red painted high mite bees. Next slide. We would also expect that those high mite bee visitations would be correlated to an increase in mite load. So colonies that were visited, click again, one more time. Thank you. I told you I have a lot of animations. Colonies that were visited by red bees should have a bigger increase in mite load than colonies that weren't visited. So if we're thinking about a receiver colony and we're looking at the percent change in mite load, we would expect colonies visited by red bees to have a higher percent change in mite load or a much bigger increase in mite load than colonies that were not visited by these high mite bees. Click one more time. And we would also expect that the robbing screens would reduce the mite load in the receiver colonies. So receiver colonies with screens should be less susceptible to visitation from these high mite bees. The high mite bees should have a harder time gaining access to these colonies. So screen colonies should have a lower mite load at the end of the study than unscreened colonies. And click again. And then if this second hypothesis is true, that receiver colonies are visiting the high mite colonies and returning home with mites, then kind of the opposite of each of those things I just said should be true. So click again. Thank you. So we should not expect to see more frequent visitations from high mite bees. The visitations from high or low mite donor bees is kind of irrelevant because the receiver colonies are the ones who are returning with the mites, bringing them home themselves. Next slide. The visitations from high mite bees should not be correlated to an increase in mites. If it's the receiver colonies who are returning home with the mites, then again, the high mite bee visits should kind of be irrelevant to the mite load. And one more time. Oh, sorry, one more time. Okay, so again, if we're thinking about the percent change in mite load, it should not differ whether or not the colony was visited by high mite donor bees if the receiver colonies are bringing home mites themselves. And one more time. Sorry, back one. Thank you. And then finally, if receiver colonies are bringing mites home themselves, then the robbing screens should have no effect on the mite load, right? If I'm going out and picking up a mite and returning home, I should be permitted to pass through that robbing screen because my nest mates will recognize me. They will let me in. I sort of have the key to the house because I live there. So the robbing screens should not have any effect on the mite load in this situation. Okay, click again. So just to summarize, I'm just going to keep referring back to this table as I walk through each one of these things. But basically, if that first idea is true, that the high mite bees are to blame, then we'll have all yeses. And if the receiver colonies are bringing home mites, then we'll have all no's. So I'll jump into results now. Click one more time. Thank you. So this is just showing the location and the proportion of high and low mite donor bee visits. So you can see that actually the majority of the visits to the receiver colonies were from low mite donor bees, from blue bees. And click one more time. And we only had 47 detections total, which when I told you we painted 16,000 bees is pretty low, but I had no idea if we would find anything. So I was pretty happy with 47. That's actually a relatively good sample size. And click one more time. And there weren't really that many visits from red bees, from high mite donor bees, only about 20%. So click one more time. And two more times. Sorry. Thank you. So when we're looking at this first thing, the proportion of high and low mite bee visits, basically, we actually saw more low mite bee visits. So this doesn't really hold up that those high mite colonies were dropping off a lot of mites because they weren't even showing up that often. So far, the receiver colonies bringing home mites is sort of the winning hypothesis. Go ahead and click. And then again, you know, when we're looking at the effect of those high mite bee visits on the change in mite load, there was no difference in increase in mite load in colonies that were or were not visited by red donor bees. So here on the left, the gray bar, we have the increase in mite load in colonies that were not visited by red bees. And on the right is colonies that were visited by red bees. And you can see the red bar looks a little bit lower, but there's no statistical difference between those bars. So really there's no difference. And certainly visitation from red bees did not result in a much bigger increase in mite load. And go ahead and click. And two more times. Perfect. Thank you. So again, this idea that high mite bee visitation is resulting in a lot of mite increases is not really holding up here. And one more time. So interestingly, the robbing screens did result in a lower mite load compared to unscreened colonies. So here on the left, we have the change in mite load in unscreened colonies, and it is a little bit higher than the mite load in screened colonies. So the robbing screens did seem to help in this instance. They did seem to slow that increase in mite load a little bit. So click again. So this is kind of a little bit of a switch here at the end. So far, it had looked like the colonies were likely bringing home mites themselves, but because the robbing screens helped here, that kind of interrupts that thinking a little bit, that maybe those colonies were not bringing home mites because, again, if they had been bringing home a lot of mites, they would have been permitted right through the screen and it shouldn't have helped. And it did help. The screen colonies had lower mite loads. So it seems like neither one of these original hypotheses we had perfectly fits the data we saw. It doesn't mean either of them are totally wrong. The red X is like a little aggressive, but it just didn't perfectly describe what we saw in this study. So, you know, I kind of started thinking about it from the other side. And click one more time, that maybe we had been focusing on the identity of the donor bees and the donor colonies, but what about what was going on with the receiver colonies? You know, it seems like whether or not the colonies were screened was important. And we also saw that colonies that received any visitor bee, a red bee or a blue bee, had higher mite loads compared to colonies that did not receive any visitor. So, you know, on the left here, that increase in mite load in colonies that were not visited by any donor bees is much, much lower than on the right in colonies that were visited by any donor bee. So go ahead and click. It seems like maybe the actual identity of the donor bee may matter less than whether or not a receiver colony was visited. And it seems like maybe any visitation is bad, and colonies who are more resistant to being visited are going to fare better. And click one more time. So what makes a colony more likely or less likely to be visited? It turned out that colonies that were visited started with higher mite loads. So here we have on the left colonies that did not end up being visited, and on the right colonies that were visited, and their mite load at the beginning of the study. And you can see that colonies that had higher mites were more likely to end up being visited throughout the study. So that kind of makes me think that maybe those colonies that started with more mites were kind of weaker to begin with, maybe less able to defend themselves against visitors, or maybe there's some olfactory cues going on that tell visiting bees that they're easier to access, something like that. But this is really kind of a bad cycle if you think about it, because if you're starting with a higher mite load and then you're more likely to be visited, you're more likely to get mites from other visiting bees, and then this just kind of keeps going in a cyclic situation where the mite load just keeps going up. So that was really interesting to see. And click one more time. And we also saw that colonies with higher mite loads received more visitors. So there's this positive correlation between a colony's mite load and the number of visitors it received. So it seems like the worse off you are, the more mites you have, the more likely you are to be visited. So again, kind of setting yourself up for a bad situation if you have a high mite load going into this time. So basically what all this boils down to is that we started thinking about this idea that receiver colony permissiveness may be a more important factor than who the actual visitors are. And go ahead and click. And that the source of the donor bees may not be as important as we thought before. And really the state of the receiver colony and how willing they are to accept these donor bees is kind of a better indicator of how their mite loads will change in the fall. So colonies that started with higher mite loads were more likely to be visited. And go ahead and click one more time. And unscreened colonies had higher increases in mite loads. And click a couple more times. So basically what I think is going on here is that we have these donor bees in the landscape, and they're looking for colonies to visit. You know, it's fall, there's not a lot of resources present, so they're looking for resources, and they may come across a strong and healthy receiver colony or a screened receiver colony, some colony that's very capable of defending itself from visitors or from robbers, and basically these donor bees do not gain access to these strong colonies. And then click again. What happens is they will actually skip over that colony until they find a weaker, unhealthy colony, or in this case, an unscreened receiver colony that's easier for them to access. And they're going to go for the weakest link where they have the highest likelihood of success. So that these weaker or unscreened colonies might be more susceptible to visitation, and thus more susceptible to these increases in mite loads and, you know, all this mixing that's going on between colonies in the fall. Next slide. So the takeaways here are that robbing screens may help interrupt mite transmission. It seems like, you know, they helped in this situation, but we definitely need to test this more. I suspect that maybe because every other colony was screened, you know, there was unscreened colonies, easier access right next door. So they maybe just gave up really easily and went for the unscreened ones. And that if every colony in an apiary was screened, they might just be a little bit more persistent. So, you know, we want to kind of play around with that in future studies. But this also has kind of interesting implications when we think about bee breeding, right? Like, a lot of us like to work with gentle bees, you know, nice kind of fat, happy, like Italian bees that are really gentle. It's way more fun to work with nice bees. But we also kind of hear all this stuff about how more defensive strains tend to have less mite issues. So we hear about feral colonies and Africanized bees having fewer mite issues. And some of the reason for that has to do with how frequently they swarm and maybe brood break stuff. But it's interesting to think that, you know, it could be because they're more defensive and less likely to just allow non-natal bees into their hives. So that's something we have to look into as well. This is all published. It was just published in March of this year. It's open access in orange there, so anybody can go and read it. If you just Google my name, there's not a lot of other Kelly Kulhaneks in the world, so you'll find it. But yeah, for anyone who's interested, you can read the paper and kind of get more detailed information. And with that, I think I'm finally done. Oh, I did just want to mention, if you're interested in indoor storage, we have this partnership with Project Apis M, where we put out a guide to indoor storage with all these different articles. We had a first edition out, I think in 2019, and we just came out with the second edition last week. And there's like lots of new information in there, including this article on wintering strategies for backyard beekeepers and potential for Varroa control and cold storage. So lots of new info. A lot of the photos I used in this presentation are from that guide, so it's really nicely produced. Project Apis M did a really good job making it look nice. And we also are actually having all of this on a website now at this URL here. And this will be updated with regular updates, kind of like on a monthly basis, new articles and new data coming out. A lot of this preliminary information I mentioned will be up there as well. So we're excited about that if you found the indoor storage stuff interesting. And okay, now I'm finally done. So I'll thank all my Maryland people, my funding sources for my PhD, and then all our Washington State people as well, and all our funding sources here and our collaborators. That's my WSU email there if you ever need to contact me or have other questions. But yeah, I think I can take questions now too, if there are some in the chat.
Thank you, Kelly. That was wonderful.
Thank you. I know it's a lot of information.
A lot of information, and you did it beautifully, and your I don't know, passion is very evident, and that's super cool. So thank you.
I like in person because I can kind of like gauge if people are following me or not. So
it's very hard to speak to just, yeah, so makes, I know the challenge of speaking without any response, but
yeah,
it was great.
Thank you. Are there any questions? I'll check the chat here if there's any questions.
I don't see any right now.
I don't see any either. Okay, great. Yeah, like I said, feel free to share my email if anyone has questions later.
Yep, Tracy. Tracy, do you want to ask a question?
Can't stand myself. Yes, please. Kelly, thanks for that presentation. I was wondering if you could email me the percentage of oxalic acid, the solution that they used.
Yes, thank you for reminding me. I will do that right after this.
And then I was also wondering if in your research, you have any data on bee mortality using oxalic acid.
That's a good question. I don't have any data personally, and I don't think they collected bee mortality data either. I think they just looked at mite mortality.
Okay, because on the Facebook blogs, it seems like people think that oxalic acid is completely harmless to the bees, and so they use it over and over and over. And because they can't see the dead bees, they think there aren't any, so they don't want to do an alcohol wash because, oh my gosh, that kills 300 bees. But I'll use oxalic acid every 4 days all summer. So I can't find any sources specifically, so I was wondering if you could help me out.
Yeah, that's a good question, and I don't know if anyone's looked at that off the top of my head. I don't have a good answer, but Yeah, I mean, I definitely would caution against treating more than necessary in any situation for sure, just because there are, I mean, even if there are an outright, you know, bee death, bee mortality, there are certainly sublethal effects of a lot of mite treatments. So that's why we recommend that you monitor first and only treat if you really need to try to reduce the chemicals you're putting into your hives as much as possible.
Wait, could you say that again? Because I preach that too. Monitor first.
Monitor first and monitor after. You have no guarantee that your treatment worked unless you monitor after. So many times we have seen that the mite load just does not go down as much as you would expect. So you have to check before and after. Yeah.
For sure. Thanks. I'll send you an email.
Perfect. Thank you very much.
I have a question about monitoring, and that is, are you careful about which part, I mean, which frame of the brood nest you're pulling from when you select your samples?
Yeah,
what's your preference? Yeah,
really big into this right now. Looking at, does it really matter where you take the sample from? I always learned the brood nest, so that's what I do. I still do the brood nest. It's cool if Randy can verify that we don't have to do the brood nest. I feel really confident recognizing a queen and have not done that on accident, sampled a queen yet, knock on wood. So I feel comfortable going from the brood nest, and that's just what I've always done, but happy to change my ways if I see some solid results.
That's what Randy said, was that the brood nest gives us a number that's higher than the average. Mm-hmm. So to go with that outer frame is the perfect average. And
yeah, I don't know though, if, you know, we have these thresholds, this like 3 mite per 100 bees idea. I don't know if that was based on the number of mites in the whole colony or samples taken from the brood nest.
That's a great point.
Yeah. So, I mean, I don't know the answer to that off the top of my head, but that's something to consider, is that the thresholds we have and the efficacy rates we have might be based off those brood nest samples.
Oh,
so
interesting. Good question. Jeff, did you have a question?
Yeah, sure. I think your mite drift research is really interesting, and I'm wondering if you had considered artificial insemination of those hives so that you would have basically clones, so you could watch that behavior more carefully.
Yeah, that's a great point. So I definitely think genetics could be a factor in this particular study. And I didn't mention this, but all the receiver colonies were new splits we made with new queens, and we got the queens all from the same breeder, and they were all Italians. So I feel comfortable that they were relatively similar genetically, but yeah, I don't know 100% for sure that they were exactly the same. That was kind of an unexpected thing that we weren't really initially looking for. So definitely eager to do it again and sort of think more about the robbing screens and the genetics and sort of what all of that looks like in practice for sure.
Cool.
Thank you.
Endless considerations.
Yes. The more I do this, the more I realize we just really don't know that much after all.
Those sneaky bees.
Mm-hmm.
Sneaky mites. But the bees are sneaky too.
Yeah, the bees are sneaky. I always say when it comes to bee research, if you need a hive to stay alive, it'll die. And if you need it to die, it'll stay alive. Like I said, one of those high mite colonies had like 10 mites per 100 bees at the start. And I had to keep doing this study until the colonies died. I thought those high mite colonies were going to die super fast. I'm like, cool, you know, I'll be done with this in 2 or 3 weeks. How long can you live with 10 mites per 100 bees? And they lived like, I did this experiment for like 10 weeks and longer. And they slowly dwindled, but it was not like a massive crash like I thought it was going to be. So, yeah.
Wow. Wow. You could tell a lot of stories, I think.
Yeah. Mm. It's fun though.
Good. Of course. It better be.
Yeah.
Good. Any other questions? Well, you were wonderful, and PSBA is grateful for you. So thanks for all your good work.
Thank you. I really hope I get to meet you all in person soon. I'll be at our big thing in Othello in the fall, if anyone's coming to that. So hopefully I'll see you there, but yeah, or before then too.
Yeah, I mentioned that. You saw that I put that in the newsletter. Yeah, so it's a gathering of beekeepers.
Yeah.
It's always a fun thing. So,
Yeah.
and
Beautiful.
camping on site in Othello, I know that facility is beautiful.
Yeah.
From the pictures I've seen, I haven't been there.
Yeah.
But.
Yeah, it's huge. A lot of potential there. It's just 2 hours away from here, so it's taking a while to get set up. But yeah, really excited to see what we make of that space. Yeah,
Great.
thank you again so much. And yeah, everybody, please feel free to email me with more questions if you think of them later. And yeah, thank you very much for having me.
Okay, thanks everybody. We'll sign off. See you in August. Watch those mites.